Now let's talk a little bit about the role of glial cells here. Remember the myelin that's on the axon? So the action potential is traveling through the axon, but the myelin actually plays an important part in providing that insulation and support. So let's take a look at how that works. It turns out that there are two types of cells that make myelin. One is a glial cell called an oligodendrocyte. Doesn't that just roll off the tongue? Oligodendrocytes. You can shorten it and just think of them as oligos. Oligos make myelin in the central nervous system. And so we can see here that there's an oligodendrocyte, and it wraps different parts of, these arms around sort of the axon and forms the myelin that way. Schwann cells form myelin in the peripheral nervous system, and they do it by wrapping themselves around the axon. So in the case of the peripheral nervous system, the Schwann cell itself is wrapped around and forms the myelin. This ends up actually being an important distinction. In the peripheral nervous system, if there is damage and an axon gets damaged or even broken, the Schwann cell, if it is able to, will form like a little tunnel and allow that axon to regrow. And that does not happen in the central nervous system. But there are cases, and it's actually fairly common for regrowth of axons in the peripheral nervous system, and that's why it relates to the fact that the Schwann cells sort of make that helpful little tunnel for the regrowth to happen in the peripheral nervous system, but that does not happen in the central nervous system. So myelin, again, super important, increases the conduction speed dramatically from 1 meter a second to over 120 meters a second. Myelin gaps within, on the axon are called nodes of Ranvier. That's where action potentials occur. And multiple sclerosis is a disease in which myelin is destroyed, and the word sclerosis refers to like scarring. And so with the loss of the myelin in multiple sclerosis, conduction speed is affected, and so it's certainly the ability to send neural signals is affected. Transmission between nodes under the myelin is by what's called local potention. And saltatory conduction is something we're going to really focus on. That's how the action potential jumps from node to node. So let's take a look here. This is the length of axon. And so in yellow here we can see the axon. And then here we can see the myelin that would normally be wrapped around it. And then there are these little gaps in the myelin. Those gaps are called nodes of Ranvier. We can see them labeled here at the bottom. So the gaps between one piece of myelin and another piece of myelin is a node of Ranvier. And what happens is, because of the myelin forming this insulation, there aren't any sodium or potassium channels underneath the myelin. They are only actually present here in these nodes of Ranvier, in these gaps. So the action potential as it travels along the axon really only happens at this point at the node of Ranvier, where there will be a whole bunch of sodium going in and then a whole bunch of potassium going out. And then underneath, the sodium that comes in floats across, diffuses until it gets here. When it gets here, it sort of crosses that threshold and makes this part of the axon positive enough that it crosses the threshold, and bam, all the sodium channels open, and then sodium will go in here, rush in, and then potassium will leave. The sodium that came in diffuses across to the next one. And so if you were to be recording the electrical potential of different nodes along the axon, you would see that the action potential would happen here at this node, and then a moment later it would happen here, and a moment later it would happen here. And in between, underneath the myelin, it's just diffusion of that sodium that came in through the action potential, diffusing across, coming over here, kicking off the next action potential. And so this process, because it hops from node to node, is called saltatory conduction. That's from the Latin, and if you happen to speak Spanish, you might know that as well. Saltar means to jump, and you can also remember that as saltatory. Kind of sounds salty, right? So sodium is a component of salt, and so that's another way to remember that. Saltatory conduction is the jumping of the action potential because sodium comes in from one node and then a moment later to the next. So you can think of the sodium jumping from node to node to node in a sense. We're coming in first at one node and then the next and then the next. And remember the refractory period? If there's an action potential here and then the sodium comes across and we trigger another action potential here, sodium could potentially float backwards, but if it comes right back here, this area will be in the refractory period, and so it won't be able to travel backwards. The refractory period keeps the neuron having an action potential here and then here and then here, only going in one direction, never being able to come back, because when it comes back, it can't do anything because that part of the axon is having its refractory period. And since we're talking about glial cells, the ones that make the myelin, we can mention that glial cells, remember, they outnumber neurons quite a bit. They are really numerous in the brain. They are really important. They increase the number of connections between neurons. So on the left, these are neural connections you see between these different neurons, and you can see the dendrites and the axons and stuff, the thinner lines. Well, there's fewer of these connections here than here in this picture. The difference? Glial cells. Where there are more glial cells, you get more connections between neurons. So they really facilitate neural communication and neural development. Other functions of glial cells includes that they provide physical scaffolds, like ladders, for migrating neurons to move along. They respond to injury and disease by removing debris in the nervous system. They provide energy to neurons, and they facilitate those neural connections, creating 7 times more neural connections when they are present. And so they are super, super important in facilitating the actions of neurons. And so, and there's even evidence that there's better performance of learning when there are glial cells that have been sort of transplanted from one organism to another to allow for, more learning to happen.